All right, so we're going to talk about mutations in this video. And so mutations are a mistake in an organism's DNA. And so they can be caused by multiple things, UV rays, certain foods have like carcinogens or how they're prepared. DNA polymerase and RNA polymerase also make mistakes, and so there can be stuff that occurs there. And so there's just mistakes that occur in your DNA. There's two types of DNA mutations. So one is a point mutation, when a nucleotide is replaced by another. So in some cases, remember, DNA is transcribed into RNA and then translated into protein. And so if you have a point mutation, you could have a change in amino acid, right? But sometimes you don't, because oftentimes in the third base, there's multiple codons that code for that same amino acid. And so you could have what we call a silent mutation. You could also have frame shift mutations. Those are when a DNA nucleotide gets inserted or deleted. And you can imagine that shifts the whole codons, right? And so that's what we call it, a frame shift. So here's a point mutation. So you see here we call wild type, or what normally occurs is CTT, and you get GAA, and so your normal is glutamate. However, if that T gets switched to an A, that's a point mutation. So here's your point mutation. And then you can see that now it's going to cause valine instead of glutamine. And I guess this is sickle cell hemoglobin or sickle cell anemia. So by changing the DNA, what happens is you change the protein, and then you can cause disease or what have you. But sometimes there's a silent mutation. So you can see here that this A gets changed to a G, but oftentimes in the third position. So here's the first, second, and third, first, second, and third. In the third position, oftentimes what will happen is, even though it changes, the amino acid doesn't change. So even though there's a mutation, we call silent because you can't see it. And so let me show you what I mean by that. So if I go back and we talk about the codon chart here, what you'll notice is that, for example, if I have CU, it doesn't matter what my third nucleotide is, right? Because if it's U, it's leucine. If it's C, it's leucine. If it's A, it's leucine. If it's G, it's leucine. So it doesn't matter. Anything that is CU something will always be leucine. So if there's a mutation here, you still will get the same amino acid. And so the fact that there's a lot of, if you look at it, there's multiple codons for each amino acid. That kind of helps guard against mutations, because even though something mutates, it might not necessarily cause a change in amino acid. You could have a frame shift mutation. So you can see here you have your shark codon ATG, and then what happens is a G gets inserted, right? Or sorry, in this case it looks like a deletion. So the G gets removed, and so now you get AAG, and so you can see everything gets shifted over one to the left, and that causes a whole new codon. And this is the worst kind of mutation, because it'll cause not only one amino acid, but you'll see that multiple amino acids after are going to be different. And so these frame shift mutations, insertion or deletion, are the worst when it comes to things that can affect the organism. And then here's some mutations. So mutations occur. They're not always harmful. Sometimes they're beneficial, and so mutations are what causes change in species.